So Human Genome Project was an international combined effort in order to sequence the genome of the human being, and the idea was to identify the genes what it contains. It was coordinated by National Institute of Health USA and US Department of Energy. Actually, Department of Energy was interested in finding out the effect of radiations on the human genome. Sequencing was performed by 20 different international organizations from all over the world. That includes United Kingdom, France, Germany, Japan and China. Obviously USA was there as a major collaborator. Project formally began in 1990s and was completed in 2003. Actually it was 2 years ahead of its schedule. It provided a blueprint for the development of a human's genome. Main objectives were to provide a complete sequence of 3 billion base pairs which are present in the DNA, and then find out the estimated genes. At that time the estimates were around 100,000, but later on we see that they are less than 30,000. The project also aimed to sequence some additional genomes of related model organisms like fruit flies, mouse and fruit fly Drosophila. The objectives also include to develop some tools or softwares to analyze the data for to derive valuable information. In the objectives, it was also included to look into the different ethical, legal and social issues. We abbreviate them as ELSI, that what are the how do this human genome information will affect into these aspects also. There were some supporting factors that contributed in the development of the Human Genome Project. So say for example in 1995, till that time the work progressed on different areas, so which helped a lot. So for example, there was the construction of genetic and physical maps of humans and mouse, and in addition to that the sequencing of yeast and worm has already been done. So they kind of supported Human Genome Project. These projects actually explored two-phased paradigms. Number one was shotgun phase, in which the genome is divided into appropriate size fragments and sequenced, and then in the finishing and assembly those fragments are put together. Before they started working on that, they conducted an initial project that was in 1995. It was proposed to work on some draft sequence genome, and that one was finished in March 1999. So what it concluded was this pilot project provided the finished genome, 99% complete genome, actually from 15% of the human DNA, and it also introduced BACs, bacterial artificial chromosomes. They were introduced as cloning vectors, so then later on they were used in the second phase. And obviously this pilot project gave a high pace to the overall project. These are some timelines and important landmark discoveries that help in the development of this Human Genome Project. So on the top we see that there are some bacterial genomes. They were sequenced first, and then yeast, and then C. elegans, Drosophila, Arabidopsis. The works also started on mouse, so there was the genetic and physical maps of mouse. They were completed, and then we also look in the human. There was the genetic maps, so actually they started in early 90s till somewhere in the mid 90s this work progressed. And the physical maps, they were also started, and then they went all the way. And then the initial final, we see that in the genomic sequencing, the pilot project which completed the 15% of the genome, and then the working draft where we have this 90%, and then in the end it finished in 2003. So there are different important achievements that have been done between these 1990s. This decade has been very, very productive. So Human Genome Project was finally announced by NHGRI, National Human Genome Research Institute, that was under NIH. Francis Collin was leading that, And then there was a commercial vendor, Celera, under the leadership of Craig Venter. They simultaneously announced it in year 2000. So knowing more about genes will definitely contribute towards improvements in life sciences, biotechnology and medicine. Here we can see the importance of this project in the sentences delivered by President Obama in 2013 State of the Union address. So he says that if we want to make the best products, we also have to invest in best ideas. So every dollar we invested to map the human genome returned $140 to the economy. So Human Genome Project aims at sequencing and characterization of human genome. It was a joint effort, mainly coming from US government. It has the institute NIH working on that, and there was a private vendor, Celera, who also contributed in it.